So thank you all once again for sticking with us for as long as you have on day 3, our final day of the IEDB user workshop. So for some of you who may have attended in previous years, you'll know that we previously only had a 2-day workshop. So this year we decided to add on the 3rd day and make it more specialized in terms of areas of research. So as you know, you've seen 3D structure analysis and how the IEDB and tools can be used for that. We've spoken about the application programming interface that we have for the database as well, if you're more bioinformatics inclined. And then we had the HIV databases as well from the team at Los Alamos National Laboratory. And so our last section of the day will focus in on CEDAR. So CEDAR is the Cancer Epitope Database and Analysis Resource. This is one of our very exciting newer projects that was started up by our team here at the La Jolla Institute for Immunology, headed up by Dr. Alessandro Sette and Dr. Bjorn Peters as well. And as you'll hear very shortly, it's really building on the foundation of the IEDB. So some of this will look familiar to you. Last year, we did present on CEDAR, and it was very much the concept phase. So we only had this project approved by the National Cancer Institute in 2021. So last year, we presented our vision of what CEDAR could look like. And this year now, we will show you an update of what has transpired since then. And we're very excited to show you that we do have a live working prototype as well. And we'll share those details with you. And now we're at 10:45, so I will move into the next section. As you may see on your agenda, this was to be presented by Dr. Zeynep Kozaloglu-Yalçın, but she's unfortunately unwell, so Alex has been very kind in stepping in to present. So you're all very fortunate to hear from our PI twice for this day. So without further ado, Alex, I will hand it over to you to present the Cancer Epitope Database and Analysis Resource.
Thank you, Nina, and our wishes to Zeynep for a quick recovery. So CEDAR, as Nina was saying, this is a new project, and you see here Cancer Epitope Database and Analysis Resource. So where cancer epitopes come from. So obviously they are derived from cancer antigens for the most part. In reality, there are also some cancer epitopes that are derived from viruses associated with cancer, such, for example, HPV, which is associated with cervical carcinoma and so forth. But those have already been curated by the IEDB because they're part of infectious diseases. But there's all this other category of cancer antigens that are encoding epitopes, and some may be derived from mutations, neoepitopes, very popular these days. There are other cancer antigens that are only expressed in tumors, and they are not expressed in normal tissues. There are other tissue-specific cancer antigens, for example, the ones that are expressed in melanomas, or for example, that are expressed in the prostate cells and so forth. And then also are other type of cancer antigens which are really linked to overexpression, like p53 and so forth. These are proteins that are also expressed normally in cells, but are really overexpressed and become targets in certain cancer types. So why do CEDAR? Because obviously this is a gap in the wealth of information related to epitopes that is available. The IEDB is funded from the National Institute for Allergy and Infectious Disease, and correspondingly has information for allergy, infectious diseases, and so forth, but not cancer. And the National Cancer Institute has stepped in to fill this gap and develop a resource for cancer epitopes. Cancer epitopes are important. They play a key role in cancer immunology and immunotherapy. There's a lot of enthusiasm for example, using cancer epitopes on cancer antigens as a mean to induce responses in a cancer vaccine, perhaps in combination with checkpoint inhibitors or CAR T cells that are specialized T cells engineered to express particular antibodies that, of course, recognize particular epitopes and so forth. And also important in understanding really the complex interplay between the host and the development or prevention of progression of the cancer pathology. There are some existing resources that list epitopes or antigens, but they do not capture the information with the same level of granularity with experiment and clinical context and so forth. Not at the same level that the IEDB type of structure does. And also conversely, there are limited resources in terms of the capacity to predict and analyze specifically cancer epitopes. So one of the point of strengths of the CEDAR initiative is that we can capitalize on 20 years of having been working on curating epitopes in the context of the IEDB and developing predictive tools in the context of the IEDB analysis resources. And these slides show the traffic and citations, so really an established resource. But most importantly, we have in place the procedures to, and the team and the knowledge to curate cancer epitopes and developing these tools. So again, we have two facets of the project. One is the database that stores the epitope information, and the other is the analysis resource that houses analysis tools and predictive tools. So how are we going to go about developing CEDAR? Of course, we need to develop a database, the ontology, the query and reporting. We can, to a certain extent, capitalize on what has already been done for the IEDB, but there are specific aspects that are cancer-specific that needs to be developed. And of course, having the database doesn't do you any good if the database doesn't have data in it. And we also want to develop aim number 3, cancer epitope prediction analysis tools, and I'll get back to that, and also implement an outreach program to engage the cancer research community, which is somewhat overlapping with the community of users that may be using the IEDB, but it's also distinct in many ways. So establishing the CEDAR database. So we applied a multi-pronged process, which is again what we have used in an iterative fashion to develop the IEDB. So we reached out to experts and asked questions about what they thought was important in the undertaking the task of a cancer database. We are generating prototype user interface and also modified the cancer curation rules and display to fit specifically the unique features of cancer epitope data. for example, capturing with different type of cancer types, detailing different type of vaccination and allo and xeno models and so forth. So this is what the current CEDAR database looks like. You can see that it's similar to the database, the IEDB landing page, but there are different things. For example, in the epitope source, you see it's a neoantigen, viral antigen, germline, and here in type of immune exposure. So is it a naturally occurring disease or an animal model or a vaccination and so forth. So just to give you an idea how this works. So for example, we may select prostate-specific antigen as our antigen, and I will get back to the issue of prostate antigens in more detail in a separate talk following this general CEDAR. But so you can put in prostate specific antigens, PSA, human, and then you can specify you want T cells, both positive and negative, but restricted to class 1 and human and naturally occurring disease. And then you will get this type of data in a similar fashion to what has been developed for the IEDB. And so if someone who is familiar with the IEDB will find this relatively intuitive with different panes for the epitopes, the antigen, the assays, and so forth. So that's where we stand in terms of the development of the database structure and query and reporting. So where do we stand in terms of curation of the data? Here we follow again the footsteps of the IEDB. As you may recall, I showed this slide also yesterday, there's a process by which there is a first very broad query that captures anything that is epitope related, whether it's cancer or not. And then a automatic classifier further refines this PubMed query and also puts the potentially curated papers in different bins related to their topic, allergy or infectious disease or cancer or transplantation. And then the potentially curatable records are inspected by reading the abstract, and this actually eliminates a lot of not relevant, non-curatable potential information. And then the ones that pass to this stage are actually curated or inspected by the PhD level curation team that reads at the paper and determine if the paper is curatable or not. And all these steps are done in a casting a broad net to make sure we don't lose information or potentially relevant papers. And that's why at each step there is a substantial drop off as the information and the potential records are scrutinized more closely and not relevant information is eliminated. So the interesting point of having this process is that at this stage, we already identified and categorized, even though we had not started to curate them yet, the cancer relevant potentially paper potentially containing cancer epitope information. And so we have somewhere between 3,000 and 4,000 potentially relevant records. And we started curation by focusing on prostate antigens and neoepitopes. And this has been going on now for about 2 years when we started doing some prototype, pilot curation, and then really in from the fourth quarter of 21, we have really increased our efforts, and you see that the slope is ramping up. So prostate cancer curation, again, was the first category that we tackled, and you see here in red the papers that are standing as a function of time, and in yellow, the counterpart, the papers that have been curated. And at this point, we're essentially done. We are 97% completed. Then we are essentially in real time. The 7 references in progress are a reflection of the fact that over time, naturally, new papers come out, and so we curate them, and we are in maintenance. And neo-epitopes, was started a little bit later, but we're already 3/4 of the way there. Now, the third goal is to provide a validated set of cancer prediction and analysis tools. The rationale for that is that we have predictive tools that have been developed and are currently housed in the IEDB. By and large, those tools have been developed with the infectious disease application or allergy application in mind. And so are not necessarily optimized for a cancer scientist community. And most importantly, also the training data was infectious disease data. And so there may be unique features that are associated with cancer epitopes. We might have a different affinity, a different nature. There is in cancer, of course, the paramount influence of the host self-tolerance and also the highly suppressive tumor microenvironment that may change or fine-tune the characteristics of the cancer epitopes. And so we want to take that into account and develop novel prediction tools for cancer epitopes with a combined assessment of expression and binding, for example, and additional features, some of the ones I already mentioned. The other point is that there are a number of tools that have been independently developed by different groups that are focused on cancer, neoepitopes, neoantigens, and so forth. And what we want to do is provide a web implementation of these published tools that are often hard to implement for, especially for the not so bioinformatically sophisticated user. And so we want to provide this as an easy web implementation access. Obviously, we can't do everything, so we are reviewing different tools, and we will prioritize based on what resources we have available and what are the order in which different tools will be also hosted in the CEDAR database. Another aspect of the analysis resource would be to assemble, leveraging the efforts in going on in the database component, a comprehensive set of cancer epitope data and make them available for bioinformaticians so that they can play with it. They can use them to train tools, test the performance of different tools. And like we do for the IEDB, we will conduct benchmark evaluations of prediction tools specifically on cancer epitope datasets. And you see here some examples of benchmark targets for prediction tools in the box below. Outreach is an important key element of our efforts. It's important for the IEDB, it's just as important for CEDAR, especially because we are the new kids on the block. So we need to make sure that we reach the community, we make the community aware of the existence of CEDAR, and we listen to what the cancer community has to say regarding CEDAR and their suggestions for improvement and optimization. So we are writing manuscripts to make the CEDAR initiative visible in the context of peer review publications, also taking advantage of the feedback of the scientific community. We are starting to go to different conferences and having booths to spread the word, and also as the opportunity arises, we also have presentations or posters, again, in a context of a community outreach. So those are the main components of CEDAR. Working prototype for a cancer epitope database, literature, going on, moving rapidly. Prostate is done. Neo-epitope will be done. Then we will move to other categories, such as, for example, MAGE, antigens, or other categories of cancer antigens and epitopes. A lot of work is going on at the level of tools and benchmarking, and of course, community engagement, a key component, because what is the work that is going on for aim 1, 2, and 3 really only makes sense in the context if a community uses these tools, these benchmarks, this database and of cancer epitopes. And with that,